Well, good morning, everyone. My name's Anthony Diaz. Today I'll be presenting on the project that I worked on during the past year, my research year in the Miami project with Dr. Levy and Shelby. And it's entitled Schwann Cell Delivery via a Novel 3D Collagen Matrix Conduit Improves Outcomes in Critical Length Nerve Gap Repairs. Okay, so about 13 to 23 per 100,000 suffer from a peripheral nerve injury every year worldwide. And in traumatic nerve injuries, a large gap may occur, as shown here, between both the injured nerve endings, and this poses a significant challenge for recovery in the sense that regenerating axons regrow extremely slow, about 1 millimeter per day. And so with Wallerian degeneration, the gap may get larger. And so joining these is very difficult. And so therefore the current standard for repair involves taking the sural nerve and to bridge that gap. And oftentimes you have to take the sural nerve from both ends depending on the size of the gap. And so as I have mentioned, the autograft remains a standard for treating these nerve injuries. However, this method has many limitations, including sensory loss at the sural nerve harvest site, and then this requires multiple incisions, differences in diameter of the injured nerve versus the graft that you're going to be using, and therefore possible lack of adequate tissue in very large nerve injuries. And so the idea kind of came out to use a different graft. And so there are two types that you can look into. There's a decellularized cadaveric tissue is one. However, introducing the neurotrophic factors that have been shown to help regenerate these axons or cellular supplementation is very difficult with these. And then there are the manufactured conduits. There are first versus second generation conduits. And these are typically collagen based. And among these, the first generation conduits are typically hollow versus the second generation, which have like an inner matrix to allow the axons to grow longitudinally. And this inner matrix is kind of axially aligned. You can see an example of the one that we'll be talking about here on the right. And this is a Neurogen 3D matrix. And so what it allows is this collagen matrix allows Schwann cells to migrate along a linear path, and then it forms kind of these linear columns to support elongation and myelination of the axons. And so there's this critical gap length that exists beyond which nerve regeneration doesn't occur within conventional axon guidance channels without the addition of exogenous growth factors. Or like a segment of a nerve tissue that you can insert within the graft. And so in rats, this is about 10 millimeters. Our model that I'm gonna discuss and that we used in our research was a 13 millimeter injury to be well beyond that critical gap length. And then in humans, I mean, this is very difficult to test, but it has been shown that potentially around 3 centimeters is where that critical gap length occurs. And so there is decades of research here at the Miami Project investigating various methods for repair and regeneration of severely injured peripheral nerves across large gaps. And these are just a few examples of recent papers that have been published in our group, and all with the goal to be able to investigate how these autologous Schwann cells implanted within a novel conduit can improve axonal regeneration across a long gap injury. And so that was the goal of our project. And so here I kind of briefly describe the experimental setup of the research that I worked on. We used male Fisher rats. They were a 13 millimeter sciatic nerve injury was created, and the animals were separated into three separate groups. We used a sciatic nerve autograft as a positive control. And then we used the Neurogen 3D conduits supplemented either with autologous Schwann cells or with saline as a negative control. And then we tested behavioral analysis. We did electrophysiology in vivo, And then we weighed the muscle, the gastrocnemius muscle to test for atrophy, and did histological and immunohistochemical analysis. And so the first step, the first portion of the project was to establish a reliable method of loading conduits and of loading the Schwann cells into the conduits. And we found that placing we use a 15 millimeter conduit inside of a 15 cc into a 1 cc syringes, filled with a Schwann cell solution of 100,000 cells per microliter, and then applying a negative pressure through that plunger produced the best results for loading. And as you can see here, a homogeneous distribution of GFP labeled SCs, the Schwann cells, is seen throughout the conduit. And this is just actually an example of a human-sized conduit that's 30 millimeters long. It was just easier to show that image. And so this is an immunohistochemical analysis that we did initially. At 16 weeks post-transplantation, the animals that were sacrificed, and some of the grafts were sectioned longitudinally for immunostaining of the neurofilaments by RT97C, and as you can see here in red, and the GFP labeled Schwann cells in green. We selected regions of interest at 3 millimeters from each end of the conduit and then at the midpoint to analyze for the density of these neurofilament signals. And you can see here by 4 weeks post-transplantation, all 3 groups have regenerating axons growing into the distal segments. Here. And so notably less neurofilament signals were seen in the conduit group alone, the conduit alone group. And so, and then here we can see strong interaction between the GFP labeled Schwann cells and the neurofilaments visible in the segments of group B. So this was a group that was obviously supplemented with the Schwann cells, and you can see that there is a strong interaction. As a matter of fact, you can actually see that the neurofilaments, there are a larger signal density of neurofilaments were observed in the 16 weeks as opposed to the 4-week groups. And these are the regions of interest that were selected for each of these sections. Okay, and then we also perform morphometric analysis on axial semi-thin sections at 4 or 16 and 16 weeks post-transplantation. And we analyzed the myelinated axon count using stereology. And then we also measured axon diameter and myelin thickness at 100x magnification. And so at 4 weeks, the autografts had a significantly greater number of myelinated axons than both conduit groups. And so you can see here, significantly greater. But at 16 weeks, the axon counts were similar between the autograft and the conduit with SC's group, and significantly greater than the conduit alone at all 3 segments of the conduit. And so this was sustained throughout the distance of the conduit. And then when we look at the axon diameters, we observed similar results. The axon diameters were not dissimilar between the conduit with SCs and the reverse autograft, but both were significantly greater than the conduit alone throughout the graft. And myelin thickness was also a measured point that we looked at, and we observed that with group B having a thicker myelination than group C at nearly all points. So at the midpoint and at the distal end, the myelin thickness was greater. And so what does this mean for the regeneration of the axons and the functional recovery of these animals, right? And so we looked at electrophysiologic testing that was done 1 day prior to sacrifice of the 16-week animals. And so what we did was the sciatic nerve was stimulated percutaneously under anesthesia through a pair of monopolar needles electrodes at the sciatic notch, and compound muscle action potentials as seen here were measured at significantly distal muscle groups, so like gastrocnemius, the tibialis anterior, and the plantar interossei. And these are just representative compound muscle action potentials of the contralateral limb, which was used as the positive control. And so then with increasing, we observe here that with increasing distance from the stimulation site, there's a mean onset latency that progressively increases in all experimental groups, differing significantly from the contralateral control at the TA and the PI. So here the onset latencies were very similar to the uninjured limb. But as the distance increases from the simulation site, that onset latency obviously increases, but it becomes significantly different from the uninjured limb. And then here we measured amplitudes, the amplitude of the compound muscle action potential. And none of the groups achieved values that were similar to the contralateral control, although the two most proximal muscles, groups A and B, displayed similar amplitudes that were also significantly larger than in B, and these were here. Okay, and so then interestingly, we performed a linear regression analysis comparing the morphological data with the electrophysiological results, and we see that increasing axon counts was correlated with faster conduction velocities and with larger amplitudes, and also larger axon diameters and thicker myelination was also strongly correlated with conduction velocity. And so then here we looked at the weight of the gastrocnemius muscle, and we can see that there is less muscle atrophy in the group with the conduit with the supplemented with the Schwann cells than as compared to the conduit alone. Okay, and then, okay. And so then the results summarized here that we were able to successfully repair a critical gap of 13 millimeters in rat sciatic nerve using Schwann cell filled Neurogen 3D conduits based on electrophysiological, histological, and functional endpoints. And there are significant improvements in the total number and density of the myelinated axons throughout the axon Schwann cell filled conduits as compared to the conduit alone, which allowed for significant improvements in nerve conduction and subsequent decrease in muscle atrophy. And the degree of regeneration was non-inferior to the reverse sciatic nerve autograft, which is the optimal control that you can use. And so the next steps for this research is to combine these, the 3D collagen matrix conduit with our other cell products, including the Schwann cells, in a clinical trial. And we have also been discussing using exosomes, which are microvessels that contain microRNA and can potentially stimulate the axons to regenerate faster and more efficiently through these conduits. So thank you, and thank you to Dr. Levy for giving me the opportunity to work in his lab during the past year, and Dr. Burks as well for his mentorship throughout this process, and everyone else that worked together with me in helping me get this project going. As a matter of fact, the project was actually just accepted for publishing with JNS. Thank you.
Thanks, Anthony. Great job.
Thanks for bringing this project home, Anthony. You really followed it through. I appreciate it.
Appreciate it.
Okay. So we're going to be talking about the women of neurosurgery, our history, contributions, and innovations. So why talk about this? It just so happens that 2020 is the 30th anniversary of the creation of the WIN subsection. We'll go into the history of that. It was not always a subsection. The roadmap for today is that we're going to highlight a bunch of outstanding neurosurgeons that just so happen to be female. So where did we begin? How did we get here? Where are we today? How does the U measure up? And why is this worth discussing? I want to thank Dr. Wang, who inspired me to look into this topic in the first place. So some basics. Once a professor in college challenged me to name 3 female scientists. And to my dismay, I could only name 2. It's sad, but that is the reality. And there was a study done in Europe that showed that several people in the study, even scientists that were polled, could not answer this question, even to name 1 female scientist. So I'm going to play this short clip. It's from the BBC. So a quarter of people are unable to name a single scientist, living or dead. So how can that be, right? I bet everybody knows who this lady is, this Marie Curie. But, you know, there have been a lot of other female scientists, and she is an outstanding one. The reason I bring this up is that there's the fact that no one can, you know, mention their contributions, change the importance of them, which of course, no. I don't think anyone would argue that, but I think that we often don't talk about this topic in neurosurgery in a way that is celebratory, because it might bother some members of the other gender that time is being spent educating the public specifically about female scientists or female neurosurgeons. Sadly, that's probably true for a few people. I hope not the large majority. But similar to the sentiment that all lives matter, some might say all scientists matter. And yeah, of course, like certainly they do, but that's not the goal today. Throughout history, women have been treated as second-class citizens, so we weren't allowed to vote, we couldn't own property, we couldn't be educated, we couldn't run for office, participate in science, or practice medicine. So we've come a really, really long way, and I think that is part of what WINS is celebrating as their 30th anniversary. So we've been tenacious, we've overcome a lot of these obstacles, and thanks to all the women that came before me today, I have the opportunities that I have, and women can be whoever they want to be more than ever before. So the goal of today is to bring to light some of these accomplishments that women in our field have made. And let's see, we'll start with the most famous. She actually directly contributed to neurosurgery with her sister, Bronislaw. They were the first to treat cancer with radiation after Marie Curie's discoveries in radium and in the concept of radioactivity. So they founded the first institute in the world that was dedicated to treating cancer with radiation together, the 2 sisters. So in fact, Marie Curie was the first woman to win a Nobel Prize. She's the first and only woman to ever win the Nobel Prize twice. So first and only person and only woman. And the only person to win the Nobel Prize in 2 scientific fields of chemistry and physics, still to this day. So that's pretty fantastic. Her Nobel Prize was initially only going to her husband, and Henri Becquerel, who discovered X-rays. Her husband, Pierre Curie, joined her research later on after she had already discovered radioactivity. A Swedish mathematician on the board for the Nobel Laureate Committee and an advocate for female scientists notified Pierre Curie that Marie was not going to be included on this award. And so Pierre immediately contested to the board and said that he wouldn't accept it without her on it. And that's how all 3 scientists ended up on that 1903 Nobel Prize for Physics, and how she became the first Nobel laureate that was female. Another interesting thing that I think it plays into my goal of today, which is to inspire the future, other women and inspire other men to inspire other women, that their father is actually the person who inspired both of these daughters to go into science. So he taught them mathematics and physics at home, and he was banned from his laboratory in Poland because of Russia taking over Poland. So he brought home his entire laboratory to his house and started teaching his children. And that's how the two of them got involved in the first place. And I have to say, my dad didn't get banned from teaching, but my dad is also a physics and chemistry teacher who taught me all about science and how to blow stuff up in the backyard, to my mother's dismay. So we have, a lot of us have our fathers to thank for inspiring us. So the next slide, these are just some pioneering female physicians in the United States. So we have Elizabeth Blackwell here in 1849. She's the first woman to even earn a medical degree in the United States. Then we have Dr. Anne Preston, who is the first female dean of all of a United States medical school. And it was actually an all-female medical school, the first one ever founded. So she advocated for her students. She demanded better learning opportunities. And she actually was able to convince another medical school full of male students to co-educate her students with them so that they could be in the same class and have better learning opportunity. So she brought them in, and the male students, they sat in an auditorium, they threw paper, they spit wads, they called names, they heckled to the female students, but they were harassment. And the male surgery professor that came and spoke bowed to the men, but not the women, to indicate he was not happy with their presence. So that's just a small, makes me very grateful for the world that I live in today, because I don't have to face heckling like that just to come to Grand Rounds. So we've come a really long way, and that's thanks to men and women that have been very supportive of equal education. So then the next thing to mention is Dr. Rebecca Lee Crumpler. She was the first African-American woman to earn a medical degree in the United States. That's in 1864. The next is a female surgeon, Mary Edwards Walker, who's pretty awesome. She was in the Civil War, she was a surgeon. So she was the first combat surgeon that was a female. And she was actually awarded the Medal of Honor, which she's the first and only female to this day to ever receive that award from the military. And she's one of 8 civilians to receive that award, still 2020 currently. She was present at the Battle of Bull Run and the Battle of Atlanta, and at one point she was even a prisoner of war. She healed and operated on countless surgeons with no anesthetic, no antibiotics, no pain medicine in a field hospital, and saved a lot of lives, made great contributions to the field of trauma surgery. Then we have the first Native American woman to receive a medical degree. I won't try to pronounce that last name, Pacote, Susan Pacote. So that's pretty exciting, Native American. And then in 1947, a big jump, but the first woman in the United States to earn a Nobel Prize in medicine or physiology was Dr. Gertie Cori, who is the same Cori as the famous Cori cycle that we all remember from med school, but it's the catalytic conversion of glycogen that earned her that Nobel Prize. So Laura Bassi, just to take it all the way back, is the first woman in the world to ever earn a PhD in science. This is in 1732 in Italy. She's an Italian woman, and she went to University of Bologna and got a degree in physics. She is also the first salaried female teacher at a university in the world. And at that time, she was actually one of the highest paid employees for several years. Pretty exciting, because that's still something we're fighting today. So she was also the first female member of any established organized scientific group, which is the Academy of Sciences at the Institute of Bologna in 1732. So if we jump forward to today, Dr. Shelley Timmons is the first neurosurgeon to serve as the president of AANS. So not only are we members of organizations, 300 years later, we're the president. of them, which is an awesome achievement that Dr. Timmons is the only female to achieve still. However, it should be noted, Dr. Timmons does not hold the title for first female president of AANS. The title actually belongs to Louise Eisenhart. You might recognize some of the people in these pictures, particularly Dr. Cushing there. So Dr. Eisenhart was Cushing's close colleague. She was a pioneering neuropathologist and a charter member of the society founded as the Harvey-Cushing Society in 1931 that became the AANS. She served as president from 1938 to 1939, and during her lifetime, she was considered one of the foremost neuropathologists in the world, and for many years considered the world expert on brain tumor diagnosis. So we have her to thank for a lot of our histopathological understanding of tumors. When the Journal of Neurosurgery was first published in 1944, she became its managing editor and served in that position for 22 years. There's actually a very interesting publication on her decisions to accept and reject each paper that was submitted to her because she kept a very thorough log. So as I mentioned, she's the first managing editor of the JNS. Under her editorship, the journal became known as the world's most outstanding scientific publication. How did this begin, though, this relationship with Dr. Cushing? So she had been an editorial assistant of Dr. Cushing's, and then during World War I, he left for military duty. During that time, she finished his book, Tumors of the Nervus Acousticus, and she was so inspired by what he had discovered and found about surgery there that she decided to go to medical school. So she enrolled in Tufts. While Cushing's away in France, she continues to do editorial work for him while doing her studies. And in 1922, she began to keep a log of all of his operative results on various intracranial tumors. This log is actually available at Yale today to peruse. Throughout her training and residency, she continued to record every single tumor treated by Dr. Cushing. She actually would do live supravital tissue analysis where she would take the tissue directly from the OR, go to a little side room, look under this microscope like you can see in the picture here, come back into the OR, and announce her diagnosis within a few minutes. Something that we all hope to return to, that quick of a diagnosis today. So during her residency, she continued to record all of these tumors by Dr. Cushing, who was an extremely busy man and prolific surgeon. She actually joined on the team, as I discussed, from 1928 to 1934. And she was at his side as the junior associate in surgery in Boston operating rooms and actually went with him to Yale. So they currently have a registry of brain tumors that has over 2,000 specimens. And when Cushing passed away, she became curator of this registry, following thousands of patients who had brain or spinal tumors. So neurosurgeons from all over the world would send her their slides of problem cases that they couldn't figure out. And she would help them decide what kind of treatment was indicated in the diagnosis. For years, young neurosurgeons and neuropathologists would also come to study the collection and learn from her. As the first editor of the Journal of Neurosurgery, acting with the advice of the editorial board, she was responsible for making many scientific decisions on the acceptance or rejection of submitted manuscripts. So her log, as I mentioned, covering these 14 years, is a record of neurosurgical progress, truly, scientific, technical, and political otherwise, that shaped our field dramatically. The next outstanding female I would like to discuss is Elizabeth C. Crosby. You could say she's one of the most distinguished neuroanatomists of all time and just so happens to be a female. Crosby earned her PhD in neuroanatomy in 1915, producing a groundbreaking thesis on the forebrain of the alligator, in which she meticulously dissected the brain of an alligator and drew what she saw. So her work remains important today. You can still buy her textbook. Most of us have seen the images in this book before as we learn neuroanatomy. So she was awarded the National Medal of Science Achievement Award from the American Association of Women, multiple awards. American Association of Anatomists awarded her. She was regarded as an outstanding educator. People would come to her rounds on Saturdays to learn from her. And in 1957, the Galen Society of the University of Michigan established a teaching award for the best preclinical teaching in the school, named after her. So just some firsts, exciting firsts in female. Dr. Diana Beck and Dr. Sophia Enoscu, who you can find contesting reports here. So I just decided to be equanimical and say they got tied for the world's first female neurosurgeon. Dr. Ruth Jacoby was the first ABNS board certified female neurosurgeon. That happened in 1961. In 1981, Dr. Joan Vaness became the first chairwoman of the pediatric section of the AANS, but this only tells a very small part of her contribution to the field of pediatric neurosurgery, which she greatly contributed to the discovery of pediatric diseases, treatments, and the founding of the whole subspecialty. So we have her to thank for the inclusion of children in neurosurgery. Dr. Alexa Canati is the first African-American female neurosurgeon. And I pictured her pictures here just because I find her expression in the bottom picture to be, very serious, thoughtful, and almost powerful. I'll say that of a surgeon just based on what she's there to do. And then you see the picture above and she's laughing with one of her patients. And I think this highlights what women can bring to the field in terms of our emotional variability and our ability to empathize and connect with our patients in a different way that she did an excellent job of. So you can be both powerful and emotional like she was in these two pictures and be an excellent neurosurgeon. In 1988, Dr. Frances Connolly was the first tenured female professor of neurosurgery. That was at Stanford. She famously walked out of her tenured position and wrote a book about it called Walking Out on the Boys. The reason she left was due to sexism, unfortunately, but then she came back and became the director of the VA. So in 1989, Dr. Deborah Benzel was the originator with 8 other founding women to create the organization of WINS, which is Women in Neurosurgery. So in 1989, there were 8 women that met at a AANS meeting. And all noted that this was the first time they had ever seen another woman at a professional national neurosurgery meeting, or even a state level one, actually. So they met up, they talked, they all got along, and their conversations that day led to the creation of Women in Neurosurgery. So in the early years, there were so few women in the field that the exhibit floor at the national meetings featured a map with the names and locations of every woman in neurosurgery that was presenting there. Those 8 is pretty hard to find out of 1,000. Dr. Deborah Benzel was the first to serve as president, as I said. So I put 1989, but really, I guess the organization officially became 1990, which is why it's 30 years today. And today we have about 219 female neurosurgeons that are practicing and board certified. So in 2005, Dr. Karen Marasco became the first female chair of a neurosurgery department. Dr. Marasco's achievements are outstanding and extraordinary and numerous, so I won't mention all of them, but she is also the first female president of the Senior Society of Neurosurgeons and served as the 100th president. In 2014, WINS became a joint section of the AANS and the CNS and recognized as an official organization. A big achievement that I think allowed me personally to interact with WINS when I was a medical student, which was one of the inspirational things that made me realize it was possible to be a female neurosurgeon and still have some of the work-life balance that I hope to have. So this is the official stance of the AANS on women in neurosurgery as of 2008. They realized that our recruitment and retention of women was significantly lower than other surgical specialties. And they realized that fairness and equality are fundamentally right and vital to the future success of our evolving specialty. So they commissioned Dr. Deborah Binzel to publish this paper on the future of neurosurgery, a white paper on the recruitment and retention of women. It had some pretty outstanding findings, but at the time that it was published, it read that no female neurosurgeon has ever been on the ABNS or the Neurological Surgery Residency Review Committee. And until that year of 2008, no more than 2 women have simultaneously been members of the SNS. We have come a long way since 2008, actually, and it's pretty outstanding to see how rapidly things are changing for us. So thanks to these women that came before me, of course. Since 2008, Dr. Timmons became the first female neurosurgeon president of the AANS. Dr. Karen Roscoe has achieved numerous exceptional things, as I said, but she's the chair of the first department. I'm honored that she was appointed to the director of the American Board of Neurological Surgeons, so remedying one of those things mentioned above. She's the first female president of the SNS. She's the author of over 139 peer-reviewed publications and chapters, and several books. And I will mention on the side that she is married, has twin children, and she is disabled in a wheelchair. So if you ever feel like you need some inspiration, you can look at what she has achieved. It's pretty impressive. So what are the numbers? It's been a pretty rapid climb. Now outnumber in college enrollment and graduation. Since 1995, women outnumber men in acceptance and graduation for medical school. Officially, this changed this year. 56% of graduates for medical school were female in 2020. 2019, it was 49%. I don't know if COVID affected that, but it's a large number of females coming into our field. So why do only 30% of the practicing physician workforce, why is that only made up of women? There's a very small proportion if 56% are graduating from medical school. So there's a lot that plays into this. There's been a lot of research into why women maybe do not practice for as long or have deterrence to their continued success in medicine. Dr. Walter Channing, a Harvard professor of obstetrics, claimed in 1822 that one of the first and happiest fruits of medical education in America was the exclusion of women from practice. So that gives you a subtle hint, not so subtle, as to the amount of resistance there was to women being in medicine. So not only do you overcome that, but then you have to do all the struggles of normal practicing of medicine. So we've made great success, huge strides. From 1950, there was only 6% females that were in the practicing physician workforce. So now we're at 36%. That's a pretty big improvement. But where do these women go if we're at 56% graduates? So it's not that women, graduate med school and then don't finish a residency or don't go into practice. The amount of practicing women drops to 36% because of years of unequal and unfair rejection from medical schools towards female applicants. This occurred for over 150 years in the United States. So if you consider all the physicians that are not yet retired and are looking at the last 3 to 4 decades of trends of female acceptance into medical school, significantly diluting the proportion of women in the practicing pool of physicians. So this will not be the case for long, and we can expect every year that this number becomes more equal. For many years in neurosurgery, women have occupied only about 10% of the residency positions, too few to be considered a minority, defined as 15% within the field, too few to create a so-called critical mass large enough to independently attract other females. This was said by Robert Spetzler on a paper he wrote on the progress of women in neurosurgery, which is an excellent paper. And a lot of women now in the WINS organization quotes that we were too few to even be a minority. That's a pretty powerful statement when you consider it, and something I read as a medical student when I was looking into what career to go into, and seeing that there was such a low number that there wasn't even a critical mass of people that could be role models for other women to potentially follow to go into neurosurgery really motivated me to become one of those women. And I think that as time goes on, there'll be more and more of us to set an example for the future. So what happens to these women once they get into academic neurosurgery? So this graph below it's from 2012, so it's improved since then, but it does show the gender breakdown just to give you a visual representation of what it looks like to be a female in neurosurgery. Obviously, we're the pink, the boys are the blue. So this paper looked at the H index for 1,052 academic neurological surgeons from 84 different institutions. There were several left off simply because they didn't have a full listing of their faculty on the website, so they couldn't do the statistics to look at the proportion of females. But overall, men had statistically higher research productivity. The mean was 13.3 for the H index than their female colleagues, which the mean was 9.5. And it was obviously statistically significant with a p-value of less than 0.0007. But when they actually broke this down and looked at academic rank as a secondary stratification, there were no statistical differences in the H index at the assistant professor level, at the associate professor level, or at the senior professor level. So what does that mean? In 2007, an interinstitutional longitudinal cohort study from the Mayo Clinic suggested that while men publish an overall greater amount of articles throughout their career, greater amount of articles eventually become women as well. They end up having higher publication rates later in life, and their research productivity increases as they get older in their neurosurgical career. So while men might be slowing down, women take some time to ramp up. They offer several reasons for this in this paper, primarily the unequal advancement of men and women in the fields of research, the unequal publication of female first author and senior authorship papers, and the lack of grant funding given to females. So there's a lot to overcome there, but as the women establish themselves and become more and more well-known, they end up being more and more successful. So when you actually break it down by their academic rank, I think this is why you see that H index is the same for both males and females. So I thought that was interesting. And the Mayo Clinic paper also suggested that maybe academic productivity shouldn't be the only thing looked at in terms of advancing young professors in academic rank. This is just a look in 2018 at first and senior female authorships in high impact journals, primarily the JNS and neurosurgery. But they noted that another interesting finding of their study was that female first authors statistically had a tendency of co-authoring with female senior authors. So this gives rise to the thought that the existence of cooperation among women in neurosurgery exists, and it's a phenomenon that is associated with mentor-mentee relationships. So thankfully, to the women who came before us, they're trying to bring us up as well. And you can see that in these numbers that are on the table. And interestingly, our percentage of first authorships is higher than our percentage of women in academic neurosurgery, which is about 6%. So how do we compare at the University of Miami? We're actually doing really, really good. In 2020, there's 219 ABNS certified female neurosurgeons, 25 full-time academic neurosurgeons, and 3 female chairs of a neurosurgery department. As of this year, 16% of neurosurgery residents are women. 3 out of 21 women is our current ratio, or 3 women out of 21 residents, and so that's a 14%. Last year we had Angela here, so that's 19%. That's awesome, above the average. Currently we have 4 female attendings, which is 20%, way higher than the national average of just females existing in neurosurgery, and way higher than the amount of females in academic neurosurgery on top of that. So we're really excelling there, and I'm excited to have so many female role models to learn from. So what are the hurdles? What are we still dealing with that's preventing the best and brightest of the future to join our wonderful specialty? This graph shows the percentage of female residents by specialty, surgical subspecialty, and in neurology here with general surgery, and clearly we are the lowest. We've been lacking for a long time. This is only up to 2004 though, so I think it's not fair to judge the state of things by this graph alone. We have made huge strides, as I pointed out. So some of the things mentioned though in these papers that looked at the lack of retention of medical students, the things that the medical students mentioned was the lack of positive female role models, work-life balance concerns, reproductive concerns because it's the longest residency. There are obvious biological constraints on women, and there's fear of retribution for starting a family. That varies, I think, once you become a resident, but that is the perception of medical students. There's outdated career and training structures, discrimination from co-residents and peers, discrimination, mistreatment from superiors, lack of desire to work with inflexible colleagues. The list goes on, but I think a lot of it is actually medical student misperception of the reality. And the reason there's a misperception is because there aren't enough female role models for them to learn from and look after and say, oh, it is possible. Oh, there isn't that much mistreatment. Oh, you do get along and your colleagues support you, things like that. So you can have children, you can have a family. So as time goes on and more women join our specialty, and that critical mass that Dr. Spetzler mentioned occurs, hopefully show the future of women that not only is neurosurgery one of the most exciting fields, but it's a field that they can be part of. So this quote by Dr. Carol Miller kind of resonated with me and with a lot of women in the WINS organization. It never occurred to me that I couldn't be a neurosurgeon, which, is something I felt and didn't realize the obstacles of the women that had come before me, what they've gone through, and then continue to persevere anyway. So what are we gonna do to correct this disparity? I wanna highlight a current female neurosurgeon, Dr. Aruna Gandhi from Northwestern. So she's made it her mission to foster women in medicine through mentorship programs. She works at the high school, college, and graduate level. She's worked with students at the Illinois Math and Science Academy, specifically female students, and has led to 66% of the students in the program pursuing a career in medicine. That's amazing. She has worked with over 50 medical students at Northwestern School of Medicine who went on to pursue a career in neurosurgery and spine surgery. So of those medical students, 9 were women, which is 18%. So she's moving the bar up. And I think what she's doing is an excellent example of how we're going to remedy this in the future. That is the end of my talk. If anyone has any questions or comments.
Catherine, that's a great job. And I think your review of historical women figures is very comprehensive. I just wanted to throw in a plug for Rita Levi-Montalcini. She was the second woman in neuroscience to win a Nobel Prize. She won it in 1986. And what's cool about her is that, she discovered nerve growth factor, which is important in peripheral nerve regeneration, but she did much of her research in hiding in the Italian mountains during the Nazi occupation of Italy. And what she was able to accomplish scientifically, looking at eggs and embryos and growth under those conditions are incredible. And then she went to WashU in St. Louis and then eventually, identified nerve growth factor and sequenced it, et cetera, et cetera. Won it in 1986, and then she lived to 101 years old. She just passed away a few years ago, but an amazing, amazing woman in neuroscience.
That's awesome. Thank you for sharing. There's a lot of women I'm learning about still. I didn't know about her. That's amazing. Thank you, sir.
Kat Morcos here. Very nice review. I'm going to put in a plug for Rosalind Franklin. I missed the first 2 or 3 minutes of your talk. I don't know if you mentioned her. Talking about Oh my goodness, crystallography, just the discovery of DNA. Watson and Crick essentially, I'm not going to say stole, but used her data. And she died very young, 37 or 38, British chemist. And it was her pictures of crystallography that allowed the discovery of the structure of DNA. And there's a whole story about, clearly in the 50s and 40s, talk about sexism. It was probably at its height, and how sexism, nobody recognized her contributions till posthumously, essentially. She, Rosalind Franklin, you may want to kind of read her biography. Yeah.
She's amazing. Yeah, I say they stole. But,
Yeah.
Thank you, sir.
So good morning, everyone. My name is Stefano Chang. I'm a graduate student here at the Miami Project, working in the labs of Dr. Noga and Dr. Guest. My talk is entitled Brain and Spinal Cord Stimulation for Gate Ignition in a Pig Model of Spinal Cord Injury. And today, by telling you about some of the work in our labs, I hope to convince you that the pig is a useful animal model for the fields of neuromodulation and spinal cord injury, that a brainstem region known as the mesencephalic locomotor region, or MLR, deserves more attention as a therapeutic target for gait disorders, and that combining neuromodulation at multiple points along the neuraxis is a strategy that could help better understand the nervous system and increase therapeutic yields. Let's start with spinal cord injury, which the National SCI Statistical Center estimates as having a national prevalence of between 250 and 400,000, and occurring disproportionately in otherwise healthy adults. It affects multiple systems, but the loss of mobility is important as it leads to reduced quality of life and many costly and life-threatening medical comorbidities, such as pressure sores, musculoskeletal atrophy, cardiorespiratory dysfunction, and even cognitive dysfunction. Neuromodulation strategies, by which I mean the use of electrical interfaces to alter or augment neuronal activity, have really shifted the paradigm of potential functional recovery after SCI. Deep brain stimulation and epidural stimulation are the two techniques that I'll be discussing today, but there are others, such as vagal nerve stimulation and transcranial magnetic stimulation. The most popular example of this in spinal cord injury is obviously epidural spinal cord stimulation, which is currently being used experimentally to restore standing and stepping, coughing, cardiovascular function, and bladder function. And this is a video from Susan Harkema's group from the New England Journal, which shows a 32-year-old gentleman, C5 Asia B, for 3 years before receiving epidural stimulation and intense locomotor rehabilitation. He's now able to walk with this. So this has really been a game changer for the field of spinal cord injury. And what it highlights really are that first, many motor complete patients must have anatomical sparing of descending fiber tracts that cross the lesion. And second, that neuromodulation and neuroplasticity through training may be able to uncover and even enhance the functionality of these spared circuits. The computational and experimental models suggest that epidural stimulation works by activating the large myelinated 1A afferents that feed into and increase the excitability of the intact spinal locomotor networks that are below the level of the lesion, and essentially increases their receptivity to the residual descending signals. So what are some of these descending pathways, and can they also be augmented after injury? So in clinical neurology, we place a lot of focus on the corticospinal tract because that's the main pathway we use for volitional movements, especially involving fine motor control of the hands. However, parallel descending control pathways do exist, and the one for controlling locomotion is phylogenetically older and based in the brainstem. So in 1966, a Russian group, Shik-Sever-Nurilovsky, discovered that electrical stimulation of a part of the midbrain in cats, which is now known as the mesencephalic locomotor region, could evoke locomotion. And this occurs through descending reticulospinal and monoaminergic pathways to activate the locomotor networks in the spinal cord. And the MLR has been found to be phylogenetically preserved in all vertebrate species studied to date, from lamprey, where it controls swimming, to rodents and cats, where it controls walking and running, and now in pigs, as well as in evidence of its existence in humans, functional imaging studies, and electrophysiological recordings. So there's some evidence to suggest that these glutamatergic reticulospinal neurons are important in functional recovery after spinal cord injury. First, there's frequently sparing of these tracts due to their distributed locations in the spinal cord. And second, inactivation of these neurons in a rodent model of spinal cord injury actually eliminates the benefit of epidural spinal cord stimulation. So this is a video from a paper by Courtine's group, which illustrates these points pretty well. So those are the reticulospinal fibers. So if these descending tracts are important in locomotor function after spinal cord injury, can we augment the activity or function of spared descending fibers using a neuromodulation strategy? So previous work in the Noga lab has shown that this is possible with electrical stimulation of the MLR in a rat model of spinal cord injury. So this is a rat with a T9 level weight drop contusion. As you can see, the hind limbs are plegic. Once the stimulation is turned on, there's immediate recovery of hind limb function and weight support such that the animal is able to walk without its harness. This figure shows the hind limb EMG activity and maximum locomotor speed in these injured rats with and without MLR stimulation. And as you can see, MLR stimulation augments the activity of EMG, as well as increases the maximum speed both before and after SCI in these rats. As I mentioned before, there's functional imaging and electrophysiological recording data that suggests that we also have an MLR. And this has attracted clinical interest in the region as a target for patients with gait difficulties and Parkinson's disease. And a number of DBS trials have actually been performed over the past 15 years or so. And as you can see, they've all targeted PPN, or pedunculopontine nucleus. And the results have unfortunately been mixed, some demonstrating small benefit to gait and others showing no benefit. So the MLR is a functionally defined entity, basically where low threshold electrical stimulation is able to initiate locomotion. Anatomically, this localizes to the mesopontine tegmentum, where the more dorsally located cuneiform nucleus and the more eventually adjacent pedunculopontine nucleus have both been put forth as potential structural correlates. In the cat, the MLR, or effective sites of evoking locomotion, partially overlap cuneiform and the dorsal pedunculopontine nucleus. And Schick and Orlovsky actually originally attributed the MLR to the cuneiform nucleus, as have many animal studies since. However, other researchers have argued that the pedunculopontine is more likely, and specifically that the cholinergic neurons that demarcate the pedunculopontine nucleus were particularly important for gait. And so there's been this debate between scientists as to where the optimal sites are for evoking locomotion, and it's been complicated by the proximity of these two nuclei. Recently, we've learned a lot more about the functional organization of the MLR with optogenetic dissection of these circuits in mice. And so this is a figure from a paper by Josset et al., which shows that optogenetic activation of glutamatergic neurons in the cuneiform nucleus had the greatest effect on locomotion, and much less so in the glutamatergic neurons of the pedunculopontine nucleus. And not shown in this figure, but also cholinergic neurons in the pedunculopontine nucleus also failed to initiate locomotion. So this is a video that illustrates that pretty well. Cuneiform stimulation evokes locomotion. This is pedunculopontine stimulation, and it has failed to evoke locomotion. Finally, a recent clinical study by Goetz et al. Looking at PPN DBS, again in Parkinson's patients with gait freezing, provides some further support for this distinction. Overall, there was significant variability in their gait outcomes, but when they grouped their patients using responder analysis, they found a cluster of good responders with significant reduction of gait. And furthermore, all these good responders that are represented here in green had active electrode contacts in or bordering the more dorsal cuneiform nucleus. The best two responders are identified here at the bottom in light green at the very dorsal range of the electrode positions. Here the authors have plotted the mean coordinates of all the good responders as an optimal target, and I've placed below that some cross sections of the brainstem showing the cuneiform nucleus here labeled CN and the pedunculopontine nucleus so that you can compare where these coordinates kind of fall. And a computational modeling study of DBS in this region found that mistargeting is not trivial here. Lead shifts as little as 1 millimeter significantly decreased target activation selectivity. And so we've actually proposed that suboptimal targeting of the MLR is an important potential reason why the DBS studies in this region to date have had such heterogeneous results. And we've devised a pilot study to specifically target the cuneiform nucleus in Parkinson's patients with gait freezing. And that study is enrolling its first patient this month. So a plug for those who might be interested in being involved. Okay, so I want to bring you back to the animal studies now and why it's important to move from a rodent to a larger animal model in spinal cord injury. The pig model is being increasingly recognized as a necessary link in translational neuroscience between the rodent studies and human clinical studies. based on similarities in size and anatomy to the human brain and spinal cord. And just grossly, you can see here that the rat brain is lissencephalic or smooth, while the pig brain retains the cortical convolutions that we see in humans and in non-human primates. I think you can see the difference in size here a little better in 3D. So you have the human brain here on the right, non-human primate, comparable size in the pig model, and then the rat brain is at the very bottom, very small. This is particularly important with regards to spinal cord injury and neurotrauma, where the differences in size, anatomy, and physiological responses to injury between humans and rodents can significantly influence the efficacy of therapies. And overall, rodents do very well after spinal cord injuries, even without any treatments. Furthermore, pigs are a well-established model for spinal cord injury, and there's significant expertise in this model here at the University of Miami from the Guest lab. And so using that model, this is our experimental setup for our project here. Pigs have a DBS electrode implanted into the MLR, and in some of the later pigs, an epidural electrode as well into the lumbar spinal cord. They're also implanted with internal EMG electrodes to record limb EMG activity. They're harnessed into this manual treadmill, and we can monitor their limb kinematics, limb EMG activity, as well as treadmill speed during locomotor trials. So this is the MRI-compatible acrylic head frame that we use for stereotactic targeting of the MLR. It's affixed here to a pig's skull. And so the pig brain is large enough that we can actually use clinical grade MR scanners to get the MRI before implantation surgery. And then we target the MLR based on anatomical landmarks. This is from the Brain Atlas for pigs. It's a sagittal slice from 4.6 millimeters lateral to midline. And as you can see here near the inferior colliculus, you see a small nucleus here labeled NC. That's the cuneiform nucleus. That's one of the targets that we targeted with our electrodes. Just keep to that, not shown or labeled here, would be the pedunculopontine nucleus. It would be sort of in the region where you see the PCS here, the superior cerebellar peduncle. And here you see a coronal slice from the atlas. You have to remember the orientation of the brainstem is such that their coronal slices are kind of like axial slices for us. And using anatomical landmarks, we try to target this region near the lateral parabrachial nucleus. And just below that again, you see the superior cerebellar peduncle. So once we calculate a trajectory for our electrodes, we advance those electrodes to the target, and then we actually stimulate intraoperatively to confirm that we're in a good location. So this is a video that shows one of our intraoperative stimulation tests. Keep in mind that these animals are under anesthesia, so we do not see full locomotor activity. But if we see hind limb movements, particularly rhythmic hind limb movements, that's pretty good indication we're in a good spot. What we are able to detect using our EMG electrodes is intraoperatively we get locomotor-like EMG activity. And this figure shows our intraoperative EMG recordings during stimulation of the different electrode contacts. We use the Medtronic 3389. So these electrodes are 1.5 millimeters long and spaced 0.5 millimeters apart. And even in adjacent electrodes here, you see a much diminished locomotor response, which speaks to the specificity of the stimulation. So similar to in humans, we perform a CT scan after surgery to localize electrodes, and we fuse this to the preoperative MRI to ensure that we're happy with our targeting. And these are some pictures of the construction of our manual treadmill, which we use for most of our locomotor experiments. I was inspired by a similar build for manual treadmills in dogs after I saw some videos. Dogs love running in these, and for the most part, the pigs like to run in them too. So let me show you what that looks like. So the first part of my project was to characterize the MLR in uninjured, freely moving pig. So the first video here shows what that looks like in a pig without stimulation. This is just a spontaneous bout of locomotion on the manual treadmill. The head here is on the right. And you can see they usually have a trot, which is a diagonally coordinated locomotion. So next is an example of MLR-evoked locomotion. So depending on the stimulation parameters, they can have quite a vigorous response. You can see that this animal is attempting to gallop. And here's a typical locomotor trial. In this trial, we've actually had 2 stimulations, you can see in blue. And in the second stimulation, I've also included the hind limb EMG activity showing locomotion. You can see that with each stimulation, there's a significant increase in the speed of the animal as the animal initiates locomotion. We can also compare the average of multiple stimulated and non-stimulated trials to show that MLR stimulation does in fact initiate locomotion, and that it increases maximum speed compared to non-stimulated spontaneous bouts. We've also found that MLR stimulation can augment ongoing locomotion. So this is the video of a stimulation in an awake animal on the treadmill, one of our older videos. I wanted to show that we could augment locomotion, so this actually uses a motorized treadmill, and we've slowly increased the speed of the treadmill until the pig is having difficulty keeping up. And then we apply stimulation to see if we can augment the speed, and you can see that the animal was hitting the back of the cage, and then as soon as we started stimulation, able to keep up with the treadmill without difficulty. For the most part, once we find a current amplitude threshold, the locomotor output is controlled by the frequency of stimulation. So here we have stimulations at 20, 30, 40, and 50 hertz, showing increases in EMG amplitudes, speed, and acceleration. And the 50 hertz trials, we actually had to cut them short because of the sheer amount of locomotor output. So the other thing is that location really matters. And here I've mapped the electrode positions from some of our experiments onto the brainstem atlas. The left side here. And on the right side here is a chat stain of a similar slice showing the cholinergic neurons, which the green box here should locate the pedunculopontine nucleus. And you can see that our successful electrode positions in green are mostly dorsal to that position. The positions here in gray are positions where we didn't detect any response. And yellow is where we observed turning type of behaviors. And orange here near the periaqueductal gray actually elicited defensive type of behaviors. So pigs are pretty vocal creatures, and they will vocalize to let you know how they're feeling. And this is an audio recording and spectrograph of spontaneous vocalizations during exploration. Mostly lower frequency and not terribly frequent. And you can compare that to vocalizations during one of our periaqueductal gray simulations. So much higher frequencies and higher rate of vocalizations. And studies on pig vocalizations have shown that higher frequencies and higher rates of vocalization indicate underlying stress. Many other studies have found that periaqueductal gray simulations can evoke fear and anxiety type responses in animals. So that makes sense. We've also found that simulations near the periaqueductal gray can trigger defensive posturing behaviors that have also been described in other animal models. So here's a video showing an example of one of these responses. And this would often be accompanied by the vocalizations that I showed on the previous slide. So to summarize this first part briefly, pigs have a large brain and a spinal cord, which make them amenable to neuroimaging with widely available clinical grade CT and MR scanners, as well as neuromodulation with commercially available electrodes. Pigs have an MLR, and we've mapped the effective locomotor sites around the cuneiform nucleus. And we've also found that nearby sites, such as the periaqueductal gray, evoke defensive type responses in the pig. So the second part of our study, which is still ongoing, is to test the effect of MLR stimulation and epidural stimulation after spinal cord injury. Our injuries are performed using a weight drop impactor at the T9 level, and then we use intraoperative ultrasound to evaluate the extent of injury, as well as postoperative testing of the MPWS to ensure injury similarity between pigs. Here you can see some of the gait kinematic changes that occur after SCI in these animals. There's a drop in hip height with loss of weight support, and the joint excursions in the hind limb become more irregular. Overall, the coordination of hind limb with foreign limbs is lost, and there's loss of abductors in the hind limbs that result in the legs often crossing over and scissoring with each other. And we see that many of these gait parameters actually improve with MLR stimulation in the injured animal. On the right here, you'll see EMG activity in the forelimbs, the first 4 traces, and the hind limbs, the second 4 traces, during a bout of MLR-evoked locomotion. The animal is partially weight supported with a sling, but you can see that the hip height is increased relative to the baseline during the duration of MLR stimulation. The range of ankle excursion also increases, as does the overall locomotor output of the animal. So here's the video of the animal with stimulation. This is the rear view of the animal, comparing non-stimulated and MLR-stimulated gait. On the left here, you can see that scissoring of the hind limbs that I mentioned earlier, which improves with MLR stimulation. Our most recent animals have also received epidural electrodes, which we place over the lumbar segments through laminectomy. This is a schematic of one of our custom dual strip electrodes that we used. And overall, we found that the rostral lumbar segments are the most effective for engaging hind limb locomotion and activity. So we are able to show that lumbar epidural stimulation is able to restore significant weight-bearing capacity in the animal after injury. So here I've shown a locomotor trial where I've tried to illustrate the effects of MLR stimulation, epidural stimulation, as well as combined stimulation. Epidural stimulation here in yellow, MLR stimulation in blue, and the combined stimulation in green. In the middle here, you also see 2 bouts of spontaneous locomotion, which have lower EMG amplitudes and relatively decreased hip height. At the bottom here, I've plotted the left hip angles and right elbow angles over time together. And so, as I mentioned before, normally in quadrupedal trot, these diagonal limbs are coordinated to move together. So you would actually expect the movement of these 2 measures to co-vary together. And we can actually calculate a cross-covariance coefficient ranging from 0, so not coordinated at all, to 1, perfectly coordinated. And you can see with the spontaneous bouts of locomotion that these 2 limbs are not well coordinated, and they result in a lower cross-covariance coefficient. In each of the stimulated trials, however, we see a much better coordination between the hind limb and the forelimbs, and that's reflected in the higher cross-covariance coefficient. So this is a video that's going to show you each of the spontaneous and then the various stimulated bouts. So with spontaneous locomotion, you can see the hind limbs have a weak and irregular movement. They're not coordinated to the forelimbs. MLR stimulation is actually able to restore coordination between the forelimbs and the hind limbs and increase the amplitude of the hind limb movements. Epidural stimulation also restores stepping with a slightly more restricted range of motion in the hind limbs. And the most notable effect of combined stimulation was the greater overall locomotor output compared to either stimulation alone. So, in conclusion, pigs have an MLR. We have characterized the effective stimulation sites around the cuneiform nucleus. Neuromodulation in the form of MLR stimulation and lumbar epidural stimulation can improve gait parameters in a large animal model of spinal cord injury. As you might expect, greater effects were seen with milder injuries and after longer recovery periods. And combined stimulation increases locomotor drive more than either intervention alone, and may be a useful approach to intensive neuromodulation-based rehabilitation after spinal cord injury. Moving forward, we are continuing to gather data in more spinal cord injured animals, which is unfortunately interrupted during the pandemic. And as I mentioned before, we are beginning a clinical trial here in Parkinson's disease patients targeting the cuneiform nucleus. So people that are interested in being involved can contact Dr. Jagged or Letitia. And I have to acknowledge a lot of people that have been involved in this work. My dissertation committee, particularly my co-mentors, Dr. Noga, Dr. Guest, the Neuroscience Steering Committee, members of the Noga Lab, Francisco, Luz, and Jan, members of the Dr. Guest's lab, especially Jimena and Pedro, who do all the surgeries and experiments with me, Dr. Solano, Johans, and Joe, who facilitate our surgeries, experiments, and neuroimaging, and of course our clinical collaborators, Dr. Jagged, Dr. Luca, as well as Ian and Letitia, who made this pilot study possible. The funding for this study received from the NIH and the DoD, as well as the NREF. Thank you, and I'll take any questions.
Thanks to Stefano. It was a great talk. I was just wondering if you could elaborate a little bit more on the Parkinson's disease study that you are involved in now.
Sure. So in Parkinson's disease, there's a subset of patients who have this freezing of gait. And unfortunately, the freezing of gait is refractory to a lot of the more conventional treatments for Parkinson's disease, so levodopa, as well as the conventional STN and GPI targets for DBS. And so these patients really have no treatment options. In the past, as I mentioned, the pedunculopontine nucleus was targeted for DBS, and unfortunately, looking over the 15 years, those results haven't really panned out. There have been a few studies that suggest a mild benefit, but a lot of the more rigorous double-blinded studies show no results. So, a lot of the clinicians have actually abandoned this strategy. Have suggested that differences between human physiology and the animal studies where this was very promising are too great. But I think based on the evidence that I've shown during my talk, the new optogenetic studies which show that the cuneiform nucleus really is the principal sort of locus of locomotor activity in this MLR region, as well as the recent clinical studies which showed anatomically that the active contacts that were more dorsal near the cuneiform nucleus had better gait outcomes, shows that good outcomes are possible with DBS in this region. And I think what we've kind of underestimated is the effect of targeting and how important the targeting is. And so that's sort of the premise for our study, is that we've been kind of suboptimally targeting the MLR this whole time. And by targeting the cuneiform nucleus, we may be able to see a more consistent result in these patients with gait outcome.
Hey, Stefano, it's Dr. Guest. I have a different question. Can you hear me?
Yes.
So could you talk a little bit about your observations regarding other physiological parameters such as the blood pressure and bladder events or bladder function that you've seen during these studies?
Yeah. So with I'll start with the MLR stimulation. frequently we will see the animals void with MLR stimulation. And this is probably related to bladder centers in the brainstem near the cuneiform nucleus. Heart rate also increases with MLR stimulation, and this is probably related to cardiorespiratory centers in the brainstem as well. And it sort of makes sense anatomically that these functions are anatomically close and functionally bound, because when you are preparing for locomotion, especially fast locomotion, you need to increase your heart rate and your blood pressure and rest rate to physiologically prepare for that. So that's what we've seen with MLR stimulation. With epidural stimulation, we've also seen voiding during a lot of these locomotor trials, and that has been seen in a lot of other studies as well. And as you know, the epidural stimulation is also being studied to improve bladder function in patients with spinal cord injury. So these are sort of other parameters that besides locomotion that could also benefit from this type of neuromodulation. Yeah.
Thank you.
Yeah, hi, Stefano, it's Dr. Jagged. It was a great presentation. I think also we want to mention the fact that, our hopes are that we will be able to better, hone in on where this sweet spot is with the cuneiform nucleus with some of the newer DBS technology that's out there, particularly, steerability, right? In prior studies, I'm not sure they had that technology. And so, hopefully if we get this electrode, in close proximity to the cuneiform by being able to steer the current into certain locations, we might be able to have, better outcomes.
Yeah, I totally agree with that. I neglected to mention, but you can see in this slide here that a large component of this study is the use of these new directional electrodes. We're going to be using the one from Boston Scientific, and I think that will help us to not only increase our likelihood of targeting the cuneiform, but also to avoid targeting nearby structures, because as you know, in the brainstem there are so many structures that are close by and passing through. So hopefully this will be able to target the cuneiform and minimize the side effects of off-target effects.
Stefano, this is Dalton Dietrich. How are you doing?
I'm very well, thank you. How are you?
I have to ask this question. Maybe some of the neurosurgeons can comment as well. What's the status of non-invasive neural stimulation at the point where you could actually do this non-invasively in a patient at a very precise area? How is that technology moving forward?
I'm not a real expert on this. I know that with spinal cord stimulation, there are non-invasive methods to target sort of large areas of the lumbar spinal cord, thoracic spinal cord, to try to help enable standing and stepping. But with the MLR, that's such a specific area. We've had troubles targeting that with deep brain stimulation electrodes. So I don't see how we could effectively target that with a non-invasive method. Other targets, maybe cortical stimulation could be amenable to non-invasive methods. But I think for deep brain stimulation.
Yeah, I would maybe just add to that about the transcutaneous stimulation that's being explored in both cervical spinal cord injury and thoracic spinal cord injury. So the transcutaneous stimulation is, it has dual frequencies. It has a frequency that's very high so that the subject doesn't feel the pain of the stimulation. And there are emerging studies that have shown consistently that people with complete spinal cord injury also respond to transcutaneous stimulation. So what's going on right now is trying to determine the relative roles of this non-invasive transcutaneous stimulation versus epidural stimulation within spinal cord injury rehab. That's a very current theme.
Thank you. Nice presentation. Congratulations.
Thank you.
Yes, Stefano, I'll just say that you've worked very, very hard. You've been productive. You've looked for the inroad clinically to what we're doing. So you deserve a lot of congratulations.
Thank you.
You know, just to add that, you know, we have a very active neuroscience program at the University of Miami and a graduate program. And actually one of our Department of Neurological Surgery faculty members, Colleen Atkins, is the director, and Stefano's in that program. And this is a great opportunity for the next generation of scientists, clinicians to interact with our clinical colleagues. So this is a great example of what Stefano's doing with our scientists and clinicians. Congratulations on this alliance that's working out really well, I think.
Thank you, everyone.
Have a great day, everyone. Bye-bye.